There are only two sessions, correct? Today's session and one more session.
Oh no. So there are two sessions that will be taken by Sir himself,
Hmm.
right? That will be different. It will be, I think it is scheduled for 2nd of September and 16th of September, which
Uh,
are two Saturdays, right? Apart from that, there are sessions, there are four sessions each that will be taken by two independent TAs, which
one
will be done every week. Like,
every
for example, how it is planned is that you have your first week's assignment that is to be submitted on 30th,
Hmm.
right? If I'm not sure, the deadline is for 30th.
Mhm.
So, what these sessions will help with is we will basically solve previous year's assignments with you in this particular session because we cannot solve what assignment is given right now, right? Because you need to be graded on that.
Hmm.
But the previous year's assignment will give you some insight on like the concepts will be similar. Maybe there are some repeating questions also in this year's assignment, which I have seen, and there are. So you will basically get an idea about how to solve those assignments, what kind of questions have previously been framed for this first week of lectures previously. And so how that will help you is it will aid in that assignment solving, and you will basically be a little bit more prepared maybe for this week's material for your examination eventually. And we what we'll be doing is we will also be addressing the queries that you have, right?
Hmm.
So if you have any query that is pertaining to this first week's session, so we will try to address that also. If we can give you the answer right away, we'll definitely do that. If there's an ambiguity or if we are not sure about those things, then like maybe I will add it to my lecture notes that will eventually be provided to you, or maybe in our next session I could answer those. So this is a separate session, right? So I don't know if you could understand that, but Sir, Professor S. Ganesh will be taking two sessions. That one will be on 2nd September. That is the first Saturday of September, right? And
Mhm.
the second one will be on the third Saturday of September. That is, I think, 16 September, if I'm not wrong.
Okay, so I will note down those two. Very good.
Yeah. So
Is it
you will.
announced? Is it announced or not yet announced?
Um. Actually, I'm not sure. I don't think it has been announced as yet. You will eventually receive some kind of a notification or reminder from NPTEL, right? I don't think the announcement has been made as yet. But this is what we as TAs had already been informed. So that is another, yeah.
I don't know whether you can see this one. I have
Yeah,
filled
yeah, yeah.
everything, and there is no 16th September. There is no session from professor. So, in other words, these are all only for NPTEL sessions. What I am showing you approximately, it is not there.
Yes.
So somebody you can or somebody can announce which are of the 2nd
Okay.
September, 16th September in advance, and also
Okay.
tell all the students that there is a
Yeah.
session from professor. Most of the
Yeah.
sessions are not there by professors. So most of the
Mm.
other NPTEL. So this is a great
Yeah.
opportunity to listen to him. So you have to little bit, help other students that there is a
Yeah.
session by professor, not just by, TS. TS are great. There is
Okay.
no doubt about you.
Yeah.
You are all doing a great job. But, this session also, because I put on my reminder, it gave me snooze and then said that there is a session between 4 and 6. That's how I came.
Yeah.
And you have at least one more. Natasha has joined, I think. SK.
Yes.
Natasha.
Yeah.
Great. So at least two.
Oh, the turnout is really less than what we even began with in human molecular genetics. The last time that I was taking one of Sir's courses, it is much less than that because anyway, eventually it decreases with as the time progresses. So that it was bound to happen, but maybe the notification was not sent out to you guys. And you were one of the few people who actually did remember from the first announcement that there was a session today. Okay, anyway, we will eventually start with the session. And yeah, let me give you a structure of what we are going to do first, right? For that, I will first have to share my screen. Um, I will just give an overview of what we'll be doing.
Sure.
Just a moment.
I will also mute my mic.
Okay. Okay. So as I was already explaining before, maybe one other person out of you joined. Okay, we have another request. Let me just admit that person. We can get back to this. Okay. So I'll actually start sharing again because I'm not sure if the previous sharing would work. I am just starting with my presentation as of now, and I'll give you guys an overview of what we are going to do. Also, since it was not clear, and I don't know if that is a common query or not, this is one of the sessions that I'll be taking. I am one of the TAs for this course. I am Arveen Kaur, and I'm one of the TAs, right? There will also be sessions that will be taken by Professor S. Ganesh himself. I'm not sure if the announcement is live yet. Probably, if it is not live as yet, the NPTEL will probably communicate at least a week prior to his first lecture, uh, which supposedly was yesterday actually. So probably next week, one of the lectures sir will also be taking on Saturday, that is 2nd of September, that NPTEL will probably communicate to you. As for this particular session, I let me introduce myself because it is the first time we are interacting actually. Yeah.
At what time he is going to take the session? Do you have any idea?
Yeah, so as per what was communicated to me earlier, his session will be from 4 to 5 p.m. on Saturday.
4 to 5 p.m. And
4 to 5 p.m.
on 16th also 4 to 5. Huh?
Yeah. I don't know if it has not been communicated to you people because there has been any changes or something like that. But initial,
I will
according
go
to
through
the
my mail.
initial plan, this
I
is
will
what
go
it
through my
was.
mail and then do that.
Yeah.
So please continue with your presentation.
Okay. Let me briefly introduce myself. I am Arveen Kaur. I am one of the TAs for this course, Functional Genomics. I am basically also taking your session because I have received this fellowship, which is the Prime Minister's Research Fellowship. That has basically provided me the opportunity to teach along with do my research because I am firstly a PhD scholar, but I am also teaching, which is also one of the requisites of this fellowship that we have got. And NPTEL has very generously basically given us a platform to communicate with you guys and to bridge that gap between these recorded lectures and the queries that you guys have, especially the queries that are posted on the discussion forum, right? So what I'll do every week is every week the same time 4 to 6 p.m. What I'll do is that I will go through the previous year's assignment for that particular week. Right. So every week you will be going through 3 or 4 lectures by sir, which are recorded lectures. And you have also received you will also receive an assignment related to whatever that week's course pertains, right? That assignment will be a little different every year, and what I'll do is that I will help solve some of the previous year's assignments, because before your due date for the submission of that particular assignment. So if in case you have any queries, any generic queries or any conceptual queries that I can actually solve for you guys, which would help solve your assignment, that we will deal with. Apart from this, I have a second set of presentation that I have prepared. Let me just introduce the title slide of that also. So it is this. Apart from that, what we will also be doing is some extra questions and concepts that basically are questions that have been asked on the discussion forum. The questions that people generally have, like, they are generally confusing statements or questions or concepts in the field. So those are some other things that we will be discussing in order to get a little bit more clarity on them. So for now, back to our first presentation. Okay, back to our first presentation, wherein we will be solving some questions. Also, since you might have noticed that the chat option in this particular meeting is not open or accessible. So what I request is that if you have any doubt, maybe because the number of people in this meeting are not very many, you can maybe interject in between, stop me. I will try to address your doubt then and there. If in future the number increases, what we can do is that we can always, I can always come back to the main window after some time, and you guys can raise hands and then ask questions. For now, interjection is cool with me. So our main aim is that everything that I am conveying is understandable on your part also. So let us begin with the previous year's assignment for week 1 for this particular course, Functional Genomics, right? So how I'll do this is that in one slide, I have basically given questions, and in the next slide, I'm revealing the answer to that question, right? So I will read the question, I will give you some time to think about the question, then only we'll move on to the answer slide. And then if at all there is any connecting concept, any concept that is related to this particular question, we will discuss that a little, and then we'll move on to the next question. So the first one last year was, which one of the following orders is appropriate with regard to organization from smallest to largest inside the human body? So these are the 4 options that are given. The first one says cell to chromosome to DNA to gene to tissue. The second option says gene to DNA to chromosome to cell to tissue. The third one says tissue to cell to chromosomes to DNA to gene. And the final option says tissue to gene to DNA to chromosome to cell. So since it says smallest to largest, I think the answer is apparent. So I hope you have already reached that answer. We will reveal it in the next slide. If we would have had that chat option, it would have been a good quiz, but since we don't have it, we'll just reveal the answer, which is the second one, right? So since over here we are talking about an organization from the smallest to the largest inside of the human body, the smallest out of all of these is the gene, right? So gene is a part of the DNA, which is a part of the segment of the chromosome. And the chromosome resides inside the nucleus of the cell, and cells, multiple similar functioning cells together form a tissue, right? So it is an ascending order, and a similar illustration had been used in the video by Sir also to explain this, that over here tissues is not mentioned, but similar types of cells or cells performing similar function will basically form a tissue, right? Then there will be a cell which will have various compartments and various organelles. One of the organelle will be nucleus, which inside it contains chromosomes, right? When we look at the chromosomes, these are very tightly bound structures. If these chromosomes were to unwind or unbind, then we will receive a long thread. This long thread is the genetic material or the DNA, and a small segment of the DNA will represent a gene. This small segment of the DNA, which represents a gene, will be the will mostly be the protein coding sequence, right? So the genes are mostly the protein coding sequences in the DNA. Moving on to the second question, we have which one of the following genomic approaches was used to first successful discovery of a gene causing human disease called cystic fibrosis? Right. So the options for these approaches given are positional cloning, gene sequencing, microarray, and polymerase chain reaction. So this was, I think, 3 of them have been talked about in this week's sessions, right? Positional cloning, gene sequencing, and PCR. So out of these 3 essentially, or out of all 4 of them, which one was used particularly for the discovery of cystic fibrosis? So since this is a gene, right, it was discovered around 1980, I think around 1989, if I'm not wrong, or around 1983. I'm very bad with the history of with remembering years. So at that time, PCR had not really come into a picture. Gene sequencing was something that was being talked about, but this particular gene was basically identified using positional cloning method. So what happens in positional cloning? In positional cloning, what used to happen was that we used to find the gene based on the markers that were present near it. So for example, if there was a cystic fibrosis gene, there would be some other trait that was phenotypically visible along with this particular disease causing trait, right? So that particular trait which was visible in all the patients was also linked to this particular gene, right? Because if, for example, we had 10 patients of cystic fibrosis and 8 of them, so this is a hypothetical example, right? So let us not even talk about cystic fibrosis. For example, I say that somebody has hemophilia and the bleeding does not stop. Yeah, I have 10 patients that have hemophilia, and out of 10, 8 patients also get a very high fever whenever they have hemophilia, right? So it is a hypothetical example. So from this, I know that, okay, these 2 conditions might be linked, right? And say that the gene that causes fever is already known, right? So from this, we can use that gene as a marker for this other gene, because we are saying that it is linked. Linked genes means that they lie very closely together. That is why the chances of them always occurring together or always being segregated together is huge, right? So we identify a marker gene and along with our disease gene, and we link them to one particular chromosome. After this, the region between the marker gene and this particular gene of interest is expanded and restriction digestion is done on it. Right? Once the restriction digestion is done on this particular fragment, we receive even smaller fragments. And these smaller fragments can individually be basically synthesized or cloned into the models to test about which particular clone is basically causing the disease. This is how they would come down to identifying candidate genes, and that is how eventually the gene will be found, right? So it is a very long process to be explained in just a question. It is too small, because individually every step could basically be explained in 15 minutes alone, because a lot of techniques are involved in this. Also, I have included one of the papers, the first paper actually, to identify the cystic fibrosis gene. The major people who were credited with this discovery were the PIs, principal investigators, Chapley Sui and Francis S. Collins, right? And this paper came out in again around 1980s. I'm not really sure about the exact date. Also, over here, you see this particular person. He was featured on the cover of the Science magazine, the Nature news in 2009. So over here, if you see Nature volume 469, July 2009, this was featured and he was on the cover of the Nature magazine. And this is the same boy who was 4 years old and using whose data, whose genetic history and hereditary history, in this particular person, the cystic fibrosis gene was actually identified. If I'm not sure, the name of this guy is Danny, and he was 4 years old when this gene was discovered using his own sample. Oh, even then, he featured on the Science magazine, and around 20 years later, he again featured on Nature News, Nature News magazine, and maybe it is actually a very interesting feature. That is why I have given a PDF link. So when I share this presentation, maybe you can go to this particular link, or maybe you can find it from the volume that I have already told out loud, and you can read. So this particular PDF basically talks about how around 20 years ago, this particular gene was discovered, right? When it was discovered, what was thought was that now, since the gene has been identified, which is causing this particular disease or disease condition, then it is not very far away that we will eventually get a way to treat this particular gene and have therapies that would basically nullify all the effects of the mutations in these genes. But what subsequently happened instead was that over 15,000, I think, mutations have been found in cystic fibrosis. And although the gene is the same, multiple patients have multiple different kinds of mutations. So it is very hard to have come up with a good gene therapy which could be basically generalized to all. So these discoveries and challenges have been basically mentioned in this article. It is a very nice one. I really recommend you to go and read it. And coming on to a third question, which one of the following options is true with regard to human microbiota? Okay, I think somebody had a question.
Am I audible? Am I audible, madam?
Yeah, you are audible.
The previous question, madam, you can address either today or in the next session. If I have a new disease, in a layman's term, what if I do? I can identify a gene causing that disease. That's what I wanted to know. Because here, this is all history, very nicely done, and everything is there. Since you are a practitioner of functional genomics, you can give a very basic idea. Yes, here is a patient having a particular type of disease, and maybe taking some tissue or some sample from them. How we go about identifying, maybe this is the gene that is causing this disease, or this is the gene that is causing this disease. Some overview, if you give in the next class, that will be great. Not necessary to address today, but I just wanted to listen to you from, general practitioner point of view.
Okay, so I have a counter question about if we are talking about a novel gene Or if we are talking about diagnosing what has already been, like, there are genes that have been discovered which are linked to various forms of diseases, right? There are even rare diseases and their genes have been found. But since these diseases are rare themselves, uh, people or generally even practitioners don't often know about if there are tests for them,
And
right? Because
I'm
they
I,
don't know
I
what
I'm
they are
asking
looking at.
you. I'm
Yeah.
asking you a general method. For example, we found some 1,000 people were identified some kind of disease, and we know that samples are available. What to do with that sample and how to see whether it is a one gene or 100 gene or 5 genes causing that disease. How to do that? This whole functional genomics course is in and around that, I know. But
Yeah.
you can, since you are practicing, you can give some outline, not necessarily that that should be the only recipe. So that kind of thing will really help us also to understand how to go about. Thank you.
Okay. Sure, sure. So
You
maybe,
can do that later, not an issue.
yeah. Maybe I can make a consolidated version of what kinds of techniques are being presently used and how efficient they are, right? Because even though we have sequencing now, uh, and we have access to, say, if I wanted to get my genome sequenced, I could, but that data needs to be read or it needs to be analyzed, right? So we have different kinds of problems now. So maybe I can make a consolidated version of that and try to explain it like
Thank you. Thank you.
briefly
We
at
can
least.
do that.
Yeah,
We can do that. Yeah.
yeah. Sure. I'll remember. Okay. So, okay, the third question that we landed to was which one of the following options is true with regard to human microbiota? So, look at the options of these questions because it might not have been the best framed question. The first one was the baby inside the mother's womb will be enriched with the microbiome. The second is the number of species or variety microbiome does not change with age in humans. The third is gut microbiota symbiosis increases as we shift towards the Mediterranean diet, or all of these. So there is a framing error in this question. As like when I was reading, I felt there was a framing error in this question. But with regard to what you have studied, which one of these do you think might be true, or do you think all of them might be true? So since I read out the options, I think you might have arrived to a conclusion, and we'll move to the answer, which is gut microbiota symbiosis increases as we shift towards Mediterranean diet. Yes, this is something that this was one of the examples that was given by Sir in his lecture as well. Also, why we sometimes if you are not sure about the answer because of how it is framed, maybe you can rule out the improbable answers, right? For example, can it be all of these? I don't think so because is this particular statement true that the number of species or varieties of microbiome does not change with age in humans? I think we had a very elaborate discussion in Sir's session about this, that how it changes with the age, right? So, is there somebody who's trying to join? Um, just a moment. Yeah, we have somebody who wanted to join, and hello, Gangotri. We were already in the middle of a presentation wherein we were discussing previous year questions, right? So I will get back to that presentation. Uh, just a moment. Yeah. So we were on the third question. We were discussing about its answer and about how, if we are not sure about any question, we can rule out the improbable statements, right? In this particular case, wherein you have to say whether something is true or false. So the second one is definitely not true. The first one, if you talk about, okay, there's somebody else who wants to join. Okay, I'm sorry for the interruption. And share a link. Yes. So the answer to this particular question was gut microbiota symbiosis increases as we shift towards Mediterranean diet. Versus if we basically have more amount of Western like diet in our daily routine, it could lead to dysbiosis of microbiota, right? So moving on to the fourth question, which is which one of the following option currently correctly identifies the limitations of forward genetics approach? So there was a session that was dedicated to forward versus reverse genetics, and there were a couple of minutes that were dedicated to the limitations of each, right? So let us go through the options, which are identification of lethal mutants, or creation of desired gene mutation, or redundant gene function, or all of these. So which one do you think is the correct option, which would basically identify the limitation of forward genetics? So if you remember the session, it will be all of these, right? Why? Because in forward genetics, what basically happens is that you first observe a phenotype. Okay, I'm unable to, right? First, you observe a phenotype, right? When you observe a phenotype, you see what trait it might be linked to. Okay, I'm sorry for the writing because I'm using a mouse. What trait it might be linked to, then you identify the gene responsible, and then you eventually come to the mutation in the gene that might be responsible for it, right? So this is the approach for forward genetics. If you are doing it this way, can you identify lethal mutations? So what are lethal mutations? They are basically mutations that if they were present, they will not let an individual survive. So generally what happens in case of lethal mutations, there are no if a lethal mutation occurs in an embryo, right? The embryo might not be able to survive. So they might not be able to grow to the level that we actually can observe a phenotype, right? There might be some kind of miscarriage that might be associated with it, or they might never come into being itself, right? So we will not be able to observe the phenotype, which will basically nullify the root altogether. Then in the second option, they talk about creation of a desired gene mutation, right? So if, for example, we wanted to study a particular trait and we know that it is linked to we don't know what trait it is linked to in a desired gene mutation. So for example, we were interested in a gene, say the ApoE4 gene, which now has been linked a lot to LDL and Alzheimer's disease, right? But say we knew about this gene, ApoE, and we did know the gene sequence of this particular gene. What if we wanted to check if disrupting this gene, or if there were mutations that were present in this gene, what would be the result of that? Right? For that, we will have to introduce or synthetically produce a genetic mutation and then see what kind of a phenotype is obtained in that particular organism in which the gene mutation has been induced, right? So that cannot be done over here, because over here we already have an organism. We are observing the phenotype first, right? It is not the last in the pipeline. Also, redundant gene function. For example, if there were two genes, both kinds of genes had the same function, such as they were both related to height of a person, right? For example, one of this gene gets mutated, but the other gene is still present, and just by its presence, the height of the individual is not affected at all. Right. So how will we actually ever come to basically know the function of this particular gene that had been mutated? So if there is redundancy in the function, it is not possible to be identified by forward genetics, right? So moving on, I have basically given a flow chart for forward and reverse genetics. In case of forward genetics, as I already mentioned, the natural variations or induced mutations are basically present. After this, first, the phenotype is selected. Whatever phenotype has to be studied, that is selected. And for that phenotype, whatever gene is responsible, or what is the function of that gene, that is identified. Sorry. While in case of reverse genetics, what happens is that the sequence of the gene is already known, but the function of the gene is unknown. So we induce mutations or alter the expression of this gene, and then we see what is the resultant phenotypic effects that it produces, right? So coming to question number 5, which says, why did Beadle and Tatum develop Neurospora as a model system for their biochemical studies? So let us go through the options. The first one is Neurospora does not display the recessive phenotype. The second is the genome size of Neurospora is close to that of humans. The third is the immediate effect of mutations can be seen in Neurospora since it can be grown asexually. And the fourth is biochemical studies are easier to perform within Neurospora as compared to other model systems. So what do you think is the answer of this question? You might recall that Neurospora is one of the fungi, right? It was used as a model by Beadle and Tatum. But why was it an appropriate model at that particular time? So the answer to that is because the immediate effect of mutations can be seen in Neurospora since it can be grown asexually. Right. So Neurospora being a fungi, it has both types of reproduction available. It can perform sexual reproduction, and it is also able to perform asexual reproduction. So, but mostly the most frequent type of reproduction that it performs is asexual, right? So if there was a mutation that was to occur, there was a mutation that was to occur in a particular parental generation of Neurospora, which suppose had chromosome number N, right? The parent of Neurospora had chromosome N. That is, it was haploid, right? If there was a mutation that was to occur in this particular N, what would happen is that when it divides, right, it will form a daughter Neurospora. And this daughter Neurospora would also have this N with the same mutation, right? Because why? Because the mutation did not get diluted. How would it have been diluted? For example, if it would have done sexual reproduction, it should have had 2N. Out of this, 1N might have been mutated, right? So one of the daughters would have received normal N and the other would have received the mutated N. Right, so there were chances that this might not have formed the offspring altogether, and this one might have fused with another another spore, and it might have led to a normal offspring, right? A normal 2N offspring. But if they are performing reproduction asexually, then this mutation does not get diluted, or it does not get a chance to not be present in the next generation, right? So the immediate effects of any mutation can actually be seen in Neurospora. So this is one of the reasons why it was a good model system. Apart from this, the genome size of Neurospora is close to that of humans. This was one of the options that was given. It is very untrue. Neurospora does not display the recessive phenotype. On the contrary, since it is performing asexual reproduction, recessive phenotype is very readily visible, right? For example, in humans, for a recessive phenotype to be visible, what has to happen? That you have to have 2 copies of that particular allele for you to actually express the recessive phenotype, right? Over here, since the desired copy number is only 1, even if you have that single copy, the recessive phenotype will be visible, right? Which is the case in X-linked disorders in male, right? That, for example, if there is a recessive, I'm sorry that I'm going off topic a little, but it is a very good example. For example, in females, we have 2 X chromosomes, right? And in males, we have 1 X and 1 Y. Say there was a condition which was recessive, X-linked recessive condition. X-linked recessive condition. Okay, I'll just write R. Okay, in the females over here, you would have to have 2 copies of this particular X-linked recessive condition to actually express that phenotype in the females. While in males, although it is X-linked recessive conditions, but since only 1 X is anyway present in the males, even if that 1 copy is present, the males would actually show that mutation, right? There are examples in cases of humans also for this, and I hope that explains why it will definitely display a recessive phenotype. Okay. So I have also kept an illustrative image of the Neurospora from this particular source, neurospora.org, which is kind of a database for all the studies that have been done on Neurospora. This particular fungi is easy to grow and large amounts can easily be harvested. Also, as I mentioned, it has a haploid life cycle due to which recessive traits show up in the offspring and genetic analysis is easier, right? Moving on to question number 6, which says an enzyme that is used to generate cDNA from the RNA template is known as which one of these? First option is reverse transcriptase, the second is endonuclease, the third is DNase, and the fourth is RNase. So which one of these is an enzyme which is used to generate cDNA from RNA template? Okay. So the answer for this is reverse transcriptase. Also, before I explain this, I would like to ask this question about what is the expansion of cDNA. If anybody could answer. Since I'm not on the main screen, you are free to unmute your mic and answer. If nobody is willing to, I will obviously give the answer.
Complementary DNA.
Yeah, so it is complementary DNA. Why I did ask this is because a lot of people confuse it with circular DNA, right? Why is that? Because in Watson and in a lot of other books, that is also written as cDNA a lot of times, right? So that this ambiguity is not present, we should always remember that when it is talked in sense of a reverse transcription, it is always complementary DNA. What is it complementary to? It is complementary to the RNA strand or the RNA template. Okay. So what is reverse transcription? If we talk about reverse transcription, what happens in this is that there is an mRNA. So it is not just an RNA, it is a messenger RNA. What is the difference between an RNA template and a messenger RNA? Is that the messenger RNA lacks some part of the DNA sequence. Is anybody aware of what that part is? I'll take a small water break.
Introns.
Yeah. So mRNAs do not contain introns, right? mRNAs are composed only of exons, and exons are the regions that contain the sequence for protein formation, right? Introns are most commonly the regulatory regions. So mRNAs are processed RNAs in which these introns have been spliced out, right? So they only contain exon regions, and these mRNAs can basically be used to synthesize a complementary DNA strand, which would only contain the protein coding regions, right? The regulatory regions will have been lost. So this mRNA, if it is incubated with reverse transcriptase and with primers such as oligo-DT, then what happens is that it will be able to synthesize a cDNA strand, right? This cDNA strand will further, if the cycle keeps running, what the cDNA will be able to do is that it will be able to amplify itself, right? So we will have basically the protein coding region of the DNA and it will be amplified. Why does this particular step needs to be done, right? So if we talk about why naturally it had to be done and how basically this reverse transcriptase was first obtained, it was obtained from bacteria and viruses, right? So what happened in case of viruses was that the viruses, there were certain RNA viruses that were present, right? Everybody might be aware of that the viruses could either be RNA viruses or DNA viruses. So if they were RNA viruses, they had to, after they basically were injected into the bacteria, they had to use the bacteria's machinery to replicate, right? So if there is a RNA virus whose genetic material is an RNA, how will it replicate in the bacteria? Because bacteria have DNA in them, right? So they can polymerize the DNA, but how will they polymerize the RNA? So for that, they had a special enzyme known as reverse transcriptase, which did what? It basically converted the mRNA to DNA, right? It did not convert, it synthesized a complementary strand of DNA along with that RNA, right? So what happened was that once this DNA was actually made, it could go and it could use the machinery that was native to the bacteria to basically polymerize this DNA and to amplify this DNA, right? So that is how this came into existence naturally. Over here, this is something, reverse transcription is something that we do before PCR basically, right? So even if we have real time reverse transcriptase, reverse transcription PCR, right? What is the principle of that particular PCR is that mRNA cannot really be amplified using PCR, right? They are very sensitive and the PCR wouldn't be successful if we had to basically only do it with RNA or mRNA, right? For that, we basically use reverse transcriptase so as to make a complementary DNA sequence, which can be thermostable relatively, and it is much more stable than RNA as it is. And in PCR, you also require it to be a little thermostable, right? So since it is a more stable molecule, the PCR or the amplification is easily doable. Okay, so question number 7. Which one of the following options is true with regard to concept epigenetics? The first option is environment-induced DNA mutations lead to changes in gene expression patterns. The second option is chemically induced DNA mutations lead to changes in gene expression patterns. The third is environment-induced changes in expression patterns of genes in epidermis only. And the final option is environmental induced effects lead to gene expression patterns changes that do not involve DNA mutations. Sorry. Okay, you can go through them. Okay, so according to me, the answer is very apparent. Let us see if you have also arrived to the same conclusion, which is that environmental induced effects lead to gene expression pattern changes that do not involve DNA mutations. Okay, so epigenetics, the basic definition of epigenetics includes that there are no DNA mutations that are involved over here, right? There will be changes in expression patterns, but DNA mutations or DNA sequence will not be changed or altered, right? So the first two options say DNA mutations lead to changes in gene expression, or DNA mutations lead to changes in expression, right? So these options are automatically incorrect because they talk about DNA mutations. And in epigenetics, we never talk about alteration in the DNA itself, right? Apart from this, the third option says environmental induced changes, which is correct, in the expression pattern of genes, which is also correct, but in the epidermis only. So epigenetics is a universal phenomenon. It occurs in all tissues and cells. It is not restricted to only the epidermis, right? So that is why we arrived to this final option, that is D. Okay, let me just admit whoever has joined. We have somebody who has just joined, and okay, I'll get back to sharing my screen. Yes. Okay. So we were discussing the answer for which concept is true regarding epigenetics, and the final one statement is true, which says environmentally induced effects lead to gene expression pattern changes that do not involve DNA mutations, right? Also, I have included this table. I now believe it might not be that visible, but let me just spell it out. So, and maybe you can follow. So over here, I've listed a few epigenetic changes, a few types of epigenetic changes, right? So epigenetic changes over here has broadly been divided by into DNA methylation and chromatin remodeling, right? DNA methylation itself alters DNA. Another kind of mutation, epigenetic changes that could be done in case of DNA is acetylation, right? Which you might have come across. Okay. Let me stop sharing and share again. Yeah. Another class of epigenetic changes that Jha et al. Have mentioned are chromatin remodeling, right? Chromatin remodeling could either include histone modifications such as acetylation, deacetylation, methylation, phosphorylation, or ubiquitination, or it could basically mean ATP-dependent chromatin remodeling. Apart from these, there are other kind of epigenetic changes. I could not really find a good illustration of all the kinds of epigenetic changes in one particular illustration. Another very common one could be gene regulations, right? Because if a gene is getting regulated, you cannot say that it is altering the sequence of the gene. Right, gene regulations could be another kind of epigenetic changes, especially some kind of gene regulations such as non-coding RNAs, for example, right, which earlier had people had thought were not really fundamental to our genome. They just made up junk DNA, but that is not true anymore, right? Their functions have been coming into light since the past few decades. Moving on to question number 8, which is which one of the following options is correct with regard to approach used for identifying the defective gene causing a particular neurodegenerative disease
We
in a
cannot
large
see the
family?
slide. Hello, we cannot
Oh, you
see the
can't?
slide.
Okay, okay.
Please.
Just a moment. Just give me a moment.
Thank you.
No problem. I hope it is visible now. Um, okay, I think I had discussed until here. And is there somebody who wants to join? Okay. Yeah. So I think I had discussed until here. And after I think the slide became invisible, I was discussing kinds of epigenetic changes, which could be DNA methylation or acetylation, or they could be pertaining to chromatin remodeling, such as histone modifications, which are listed over here, and ATP dependent changes, chromatin remodeling as well. Right? So after this, I had just started with this question, which said, which one of the following options is correct with regard to the approach used for identifying the defective gene causing a particular neurodegenerative disease, considering that it is a monogenic disease in a large family? So if we had a large family, we would probably be talking about family history, right? So if we are tracing a monogenic neurodegenerative disorder in this particular family, which would be the correct approach? The first option is CRISPR-Cas9 approach. The second option is forward genetics. The third option is population genetics. And the fourth option is reverse genetics. So which one of these do you think will be the correct approach in this particular scenario?
Population genetics.
Okay, no.
Because
So since we are talking about a particular family, right? We are talking about a
forward
large
genetics.
family.
Okay.
Yeah, it will be forward genetics because over here we will talk about disease history of the family itself, right? If we would have basically written that of South Asian population or of Caucasian population, it will probably pertain to population genetics. But over here we will talk in regard of forward genetics. Why? Because in case of a large family, what is the good thing that we will basically get is a lot of family history, right? So family history will help us probably draw a pedigree, right? Through that pedigree, we will be able to identify some candidate genes or loci that might be recurring multiple times in the family, which might actually be leading to a lot of monogenic neurodegenerative disease cases, right? Over here, the explanation that was given for this question is something that I have pasted over here, which is forward genetics is a molecular genetics approach to determine the genetic basis responsible for a phenotype. And we have discussed in our previous questions also that in this, first we will observe the phenotype and then we will basically deduce from that phenotype which particular gene might be responsible for it, right? Over here, a pedigree will really help. Moving on to question number 9, which says, okay, just a moment. I'm so sorry for the interruption. That was an emergency call. Okay. So coming back to the question, which says, which one of the following options correctly refers to concept metagenomics? So metagenomics is one of the concepts that Sir had touched upon. So which one of this correctly explains it? Is it an evolving hypothetical concept in the area of functional genomics? Or it is large-scale sequencing of the genome of a large number of microbial species from the native environment? Or it is a microarray-based analysis of the species that undergo metamorphosis? Or is it all of these? So you can think about it. Just a moment.
Large-scale sequencing of the genome of a large number of microbial species.
Yeah, so that is correct. Okay, my slides are actually stuck. Now it's fine. Okay. I hope my slides are visible.
Yes.
Okay, sure. We can go ahead. So yes, metagenomics in turn does explain like this particular statement just correctly defines metagenomics, which is large-scale sequencing of genome of a large number of microbial species from the native environment. What metagenomics directly means is it is a direct genetic analysis of genomes contained within an environmental sample. So, for example, if we would talk about genomics in an individual or my genomics, what we'll talk about is a complete gene sequencing, a complete DNA sequencing of my genome, right? But if I talk about metagenomics in an individual, we would not just sequence their complete genome, which could be taken from any cell of the body. Along with that, the microbiota will also be extracted, right? And they will also be sequenced. So that refers to as metagenomics. And the final question, the 10th question, which I have also seen is in your current assignment, is the construct given here in the schematic would help in generating which one of these particular mice model. Will it create a global knockout mouse, or will it create a conditional knockout mouse, or will it create a inducible knockout mouse, or could it create any of these mice? So over here, let me just go through the construct once. The first one is one of the sequence of a gene which has 4 different exons, 1, 2, 3, and 4. Right? This is one gene that is mentioned over here with 4 different exons, and it has been logged over here. On the right hand side, we have a Cree, that is basically a region that is identified by Cree recombinase, which helps in recombination. And along with it, it has a astrocyte specific promoter attached to it. So what kind of mouse model will this particular construct give? Okay, let me just come to the answer, which is conditional knockout mouse. How will it give a conditional knockout mouse? What is a conditional knockout mouse, right? So in a conditional knockout mouse, there is not a knockout in the entire body, right? Over here, suppose we say that astrocyte specific promoter might be present. So are you guys aware of what astrocytes are? Like, I will explain what astrocyte is. A specific type of brain cell. Okay, it is a glial cell present in the brain. And what astrocyte specific promoter will do is that it will only attack the glial cells which are the astrocytes, and only in those glial cells will it cause this particular knockout, right? So that will help give a conditional knockout. I have also kept a slide of what different kinds of knockouts are, right? So over here, for example, in the first case, we talk about a global knockout, in which in the embryo itself, we have basically introduced some kind of a construct due to which whatever desired gene we were targeting has been knocked out, and there was no specific promoter attached to it, right? So what happened was that all throughout the body, that particular gene was knocked out. So we have a knockout animal that has been created, right? On the contrary, in the second illustration, what we can see is that at the embryo stage, we had introduced a construct, and this black basically represents a knocked out region. So that construct specifically knocked out some particular tissue, right? In that particular, knocked out that particular tissue means that that particular gene had basically been knocked out in one particular tissue. For example, over here, we can say that maybe in the abdominal tissue, it was knocked out, right? So it will grow as a normal mouse, except for the abdominal region wherein that particular gene has been knocked out. So this will be a cell-specific type of conditional mutation, right? Then there could be another kind of mutation in which, again, we took an embryo and we introduced a construct into it, right? This construct is present even in the second case, right? But it is not active. That is why it is not depicted as black over here. This construct is such that if we externally basically give some kind of a chemical induction or some kind of an induction from outside of the body, only then will it actually be knocked out, right? So this is also a kind of condition that we are putting. So it is also known as a conditional mutant, and this is also cell type specific. Why? Because it is also expressing over here in the abdominal tissue only. And along with these two things, it is also inducible, right? For example, we have TET-on, TET-off systems, right? That basically help produce inducible knockouts. Or we have tamoxifen inducible knockouts, right? So what will happen in those knockouts is that you will basically introduce a construct which will be susceptible to tamoxifen, right? That construct will be embedded in the genome also, but it will not work or it will not knock out the specific gene until and unless tamoxifen is actually fed to that particular model, right? So if we feed tamoxifen from outside, the tamoxifen will be reached in the body, in the blood, and that will basically lead to switching on of that system, and it will create an inducible conditional knockout, right? I have just used another illustration to explain this better, wherein a constitutive knockout mice, which is the same as a global knockout mice, will basically have knockout of that of our gene of interest throughout the body, right? A tissue-specific knockout will only have the knockout in certain targeted regions of which the promoters will be present in that particular construct. And the third is inducible knockout mouse, which will have a gene of interest and it will be introduced using a construct, but only when an inducer is also given will the gene of interest expression be lost, and thus the system is inducible. So that is all for the previous year part of presentation. We will be moving on to our conceptual slides right now. Maybe I'll stop sharing this one and share that again so that there's no glitch in between. Okay, sorry. Yeah. I hope that if anybody had any doubts regarding this or any suggestions, you might have already given it, or you are free to give it right now also. And I can see that there's a little bit of lag. I hope that is not interfering much. Okay, so if nobody has anything to say, we can actually jump on to our second presentation, which will be the lecture for week 1, right? Which okay. So, since a few of you joined in between, I would like to tell you people again that what we were actually discussing right now was the week one's assignment that was provided to the learners in the previous year or the previous session. That is what we were discussing, and I hope that the concepts related to that week's assignment would actually help you to solve the current assignment also, because that is something I cannot discuss until that has been submitted by you guys. So that is the activity we did first. Now I have a second set of presentation in which I have basically gone through the discussion forum and the doubts that were posed by different people on that discussion forum, and I have explained those doubts. Along with that, from my end, I have kept some extra questions and concepts. It is a very brief presentation, but I hope it will help. So moving on with this presentation, my first query that I actually wanted to resolve was, what is genetics versus genomics? Like, is there even a difference between genetics and genomics? Right? So I don't know. When I first was learning about functional genomics in my first year, in my first semester of PhD, this was one of the questions that actually did trouble me. So my aim over here was to actually solve that doubt if anybody had it. So what is genetics? Genetics is the study of hereditary traits, right? While what is genomics? It is the study of the entire genome or the genetic material, right? I'm not sure if just the definition of these two fields would be suffice to clear your doubts. But let us go through other points and maybe that will help you understand better. So the second point for genetics is that it involves the study of a single gene, while what genomics does is that it involves the study of interactions between genes, right? So if we are talking about genetics, we will be talking about a single gene at a time. In genomics, we will be talking about a network of genes or the interactions between the genes. In genetics, it involves the study of only the coding region of the DNA, right? So genes, what are genes by definition only? Genes by definition are regions of the DNA which basically are responsible for protein formation, right? While genomics involves the study of both the coding and non-coding regions of the DNA, right? So the non-coding region of the DNA is actually the larger part. It is more than 98% of our DNA, right? So it involves the study of the non-coding region as well as the coding region. Also, I have mentioned about what a gene is, which I just also said out loud, and what is a genome. The genome is a complete set of genetic information of an organism. Why do we say it is a complete set of information? Because not only the coding region, which is just 2% of the entire genome, it also talks about the non-coding regions, which is more than 98% of the entire genome, right? Also, interestingly, I don't need to mention it over here, but this is one of the differences that was once asked to me in my third year of undergrad, right? So in our practical examination, this is what my professor asked. What is the difference between a gene and a genome? And although I knew the definitions, I was not very sure about what it was. So I hope this is a concept that you understand. Okay, moving forward with introns.
I have a doubt.
Yeah.
So you mentioned that genome includes complete set of genetic information of an organism.
Yeah.
So it includes both chromosome and mitochondrial DNA.
Yeah.
So the second thing is the genome did not mention whether it is a human genome or any other species within the human. So whether it
Okay, so
includes all the species, whatever is there inside the human,
genome is actually
bacteria,
specific.
virus.
There could be a viral genome, there could be a bacterial genome, there could be a yeast genome. So genome is specific of whatever organism we are talking about. Right? Over here, for example, if we had to over here, we were just discussing the fields, that is genetics and genomics. Right? Uh, Say that we were discussing these with regard to mouse. Okay, so the mouse genome is a complete set of genetic information of that particular organism, which is the mouse.
Okay.
On the contrary, like we just went through another concept, metagenome, right? Metagenome would also, if we are still talking about that particular mouse, along with the genome of that particular mouse, it would also include the genome of whatever is present within its gut, whatever other forms of living beings are present within the body of that particular mouse. That would even include the bacteria, the probiotics that are the symbionts, probiotic bacteria that are present within us. So that would be metagenome, right? But when we talk just about gene or genome, we are talking about a particular organism. Okay. So why do I jump into introns after genome is because I came across one of the questions on the discussion forum wherein somebody, I think, had asked why introns are not do not code for genes. If I'm not wrong, I'm not really. I don't recall the question. It was something like this, right? So I just wanted to get this concept across because it is baseless of any one of us also to presume that somebody who's attending the course might have done all these other courses such as genetics or maybe cell biology, right? So there might be people who might be new to biology altogether. So for them, what introns is that they are regions of the gene. They are regions of the DNA segment which do not code for a particular protein, right? So they were previously known as non-coding regions of the gene only. Now the term intron has been given. Why this particular term has also been coined is because if you can just focus on this first line, right, you will see a few regions. One is the untranslated region or the UTR, which is present on both the ends. The red region, which is a coding sequence or the exon, right? It is also present over here. And between them are the introns or the non-coding regions, right? So these non-coding regions are present in the DNA. They are present in the pre-mRNA, that is the unprocessed mature messenger RNA, right? But when this messenger RNA is matured or when the processing is complete, these regions are basically deleted, right? They are spliced out or they are deleted or removed, right? So that is why they are basically like intervals between coding regions, such as this particular exon and this particular exon and this third exon, right? They are intervals between these regions, thus they are known as introns. Once the pre-mRNA gets matured, these regions are lost, and what we receive is a complete protein coding sequence, which is known as the mature messenger RNA. What this will do is it will undergo translation to basically make a protein or to basically instruct for the synthesis of a protein, right? Also, what is the function of an intron, right? So if we are talking just about an intron, was it just an interval that was separating the exons? Why is this interval present at all, right? And why does it constitute the major part of our DNA also? Like I just mentioned that the non-coding region is more than 98%, right? So why is it present at all when our DNA could just have been 2% of what it is? It might have been easier to carry, to replicate, right? But why is this present in between? It basically signifies that it might have some importance, right? We might not have understood that relevance or significance completely, but it does have some significance. So whatever we have actually understood, I will explain over here. These introns, they have important regulatory regions, right? So they will regulate the expression of the genes. They are also responsible for transcription initiation and transcription termination. For example, if we talk about a DNA segment, for the transcription initiation, the machinery for the initiation has to be recruited. Like if the polymerase has to come and bind, the sigma factor has to come and bind. There are multiple other transcription factors that have to come and bind to a region. And after that, it will start basically polymerization of that particular region of DNA or transcription into mRNA, right? So for that particular transcription to actually occur, the machinery has to be recruited. This machinery is often recruited on an intron that is lying nearby, right? So they have certain specific segments that this machinery identifies. They come and sit on it, and after that, they begin the coding of that particular DNA segment that is ahead of it. So it is responsible for both initiation and termination of transcription. Also, along with this, it contains non-coding RNAs, whose functional role has started to emerge recently. So we were talking about non-coding RNAs. For example, even anything that is not coding a protein has been termed as non-coding, but it is not that they do not have any role whatsoever, right? Like, for example, when they were a part of the DNA sequence, they had regulatory regions that were important, right? Even non-coding RNAs have some functions. Sometimes they themselves act as transcription factors, right? The roles of these particular mcRNAs are emerging, and cancer is a very hot field wherein there are a lot of non-coding RNAs whose roles are actually emerging. Also, they aid in alternate splicing and hence in generation of multiple proteins from the same gene. So I hope that you guys are aware of what alternate splicing is. So what alternate splicing says is that suppose there is one exon, second exon, and a third exon, right? So forgive me for whatever is happening apart from the square that I'm making. Suppose there are these three exons, and in between them, there are these introns that are present, right? So it is not important that all three exons actually have to join to make a particular protein, right? Sometimes what happens in alternate splicing, or what rather is the beauty of alternate splicing, is that sometimes maybe only let me number these exons. Forgive me. So I'm in a neuroscience lab, and I tend to jumble axons and exons. Okay. So suppose there are these three exons, 1, 2, and 3, right? Sometimes what will happen is that only 1 and 2 will actually be present in the mature mRNA. Okay. Sometimes what will happen is only 2 and 3 is present in the mature mRNA. So what is the significance of alternate splicing? If 1 and 2 is present, suppose protein A is being formed. Protein A is being formed, right? If these two are present, maybe an enzyme B is being formed. So from one segment of protein coding region, right? From one gene, maybe two different kinds of things can be formed depending on what kinds of introns are being removed, like when the introns are removed, what kind of splicing is occurring? Splicing is to basically get these exons together, like, okay. So what kind of splicing is occurring may basically tell what kind of protein product or what kind of enzyme product might be formed. So one gene might be responsible for, maybe if that particular gene has say 40 exons, it might be making like there are so many combinations possible with 40 exons. It might be making 150 different proteins, one particular gene, right? So that is why even though only 2% of our entire DNA is coded or is composed of genes, it can still make maybe thousands and thousands of proteins and enzymes because of this particular mechanism, alternate splicing, which is only possible because of presence of the intron itself, right? So moving on. Okay. So this question will be easy for you if you were paying attention when I was basically going through the illustration in the first intron case, in the case of the intron. So does an exon only contain one coding region? Only contain coding region, not one coding region. Does it only contain coding region? So does anybody have an answer for this? Say this is one of the regions of the DNA. This is a DNA segment. The green portion is the exons, and these portions, the blue portions or the blue line is the introns. So my question is, does the exon only contain of the coding regions? That is, coding region is just the green part.
Yes, coding region only.
Okay. I will have a water break. If anybody else wants to answer, they can. Okay. So what I can explain over here is that this is one of the concepts that is not very clear or it is not very clearly understood or even taught, right? So no, exon does not only contain the coding region. Look at this, right? Also forgive me for this particular gap. These are continuous, right? I'm not talking about exons or I'm not talking about introns over here, right? It definitely does not contain introns because we are talking about exons. But exons do contain the 5-UTR and the 3-UTR. What are UTRs? They are untranslated regions. If something is untranslated, it is definitely a non-coding region, right? But exon does contain 5-UTR. This will be one exon. Let me just mark it. This will be the first exon. This will be the second exon. And this will be the third exon. Right? So the exon does not only contain the coding region, along with that, it does also contain the non-coding region. So this is an illustration that I made. Let us go back to the one that I showed you earlier. Right, this one. So if you can see over here, this is listed as an exon which contains both UTR, untranslated region, and coding sequence. Right? Similarly, this one is an exon, and the final exon again has both the coding sequence and the untranslated region. So this like I have not mentioned yes or no over here. So if anybody needs to know, no. Okay. It also contains UTRs, 5-UTR and 3-UTRs. Moving on to the next. Okay, so for explaining that only I had kept this particular illustration. Over here I can actually explain this better. Both these things. even splicing and UTRs. So if we talk about a DNA segment, it contains UTRs, exons, and introns, right? If we talk
Excuse
about
me.
a pre-mRNA, yeah,
One small question.
yeah,
You have exon 1, exon 2, exon 3. Is there shuffling possible? Like
yeah,
exon 3 will
shuffling
come
is actually
first and then
possible.
exon 2 and then exon 1?
Shuffling is possible, but I would actually not like to give an absolute answer because once I am actually saying it is possible, I cannot think of any examples in my head. So I will refrain
Is it
myself from
possible
actually
with
answering this.
immunogen?
Yeah, it is
Is it
possible with
possible
what?
with antibodies? Is it possible with antibodies? Immune antibodies.
The shuffling of each
Empirical
of the
shuffling.
reactions.
Yeah.
Uh, are you talking about antibodies or are you talking about VDJ segments?
Yes, VDJ segments, which is required for
VDJ
IgM.
segments. I am not sure if it is the case in like in VDJ. I am not sure if it is possible or not. And if at all it is possible, I would really have to check and respond. I don't want to give an incorrect answer. So I will get back regarding this in the next lecture, maybe.
Okay, thank you.
Okay, so this illustration basically again represents that yes, even the finely spliced mRNA does contain both the UTRs apart from the exons, right? So no, it does not just contain coding sequences. Then this was one of the questions that somebody had asked on the discussion forum again. What is Bermuda principles? So this particular, I think, term was mentioned when there was talk about Human Genome Project, right? So what is Bermuda Principle is basically a set of rules that were put in order to rapidly release the sequence data obtained from Human Genome Project, right? And owing to this, the Human Genome Project scientists made every part of the draft human genome sequence publicly available shortly after production. So why is it called Bermuda Principles? Is because there were 3 meetings for this particular whether the data should be released or not. And in the first meeting itself, it was decided that they will make it publicly available. Why? Because it will help in the betterment of the society and in advancement of science, right? So it was decided in the first meeting itself. And these 3 meetings, all of them were held in Bermuda, right, which is an island near Mexico or US. Uh, and these meetings were held over there. And in the subsequent meetings, there were no new principles that were basically added to this. As far as my knowledge goes, you can basically search over here on National Human Genome Research Institute project page from wherein I have gotten this information also. So it basically gives a fact sheet about Human Genome Project and what these principles wanted to basically convey was not just the release of the sequence data, but the fast release of the sequence data. So what these scientists had decided, or these people that met had decided was that as soon as the sequence will be known, it will be released publicly. And like daily, it had to be released. Whatever was known had to daily be released or updated publicly on public forums. Then this was another question asked. So I think there were these 3 questions asked by the same person. The last discussion forum, and since it hadn't been answered yet, I did answer it over here. Why is reverse genetics done only in model systems? So I think if you are aware about or clear about the concept of reverse genetics, you will be able to answer this without me explaining, right? But since other people might also get access to this video who might not be aware about this particular thing, I will explain. So what does reverse genetics do? First, let us just concentrate on the illustration. In reverse genetics, we know about the gene, right? We know about the gene, which is a part of the DNA segment. We might know the sequence of this gene as well, right? Whatever amino acids or bases it has, that sequence also we might be aware. So from this information, we want to know what does this gene actually do? So maybe if we think about creating a mutation in this gene, such as a nonsense mutation, which will not help whatever this gene was coding be produced, right? If we create such a mutation, maybe using CRISPR or any other such technique, then we basically introduce this mutation in the genome of an organism or a model system that we have chosen. For example, a mouse over here. And then what will we do? We will see what happens to this mouse. What kind of phenotypic traits is it showing different to a normal mouse, right? So if we are manipulating any gene, right, the manipulation can only be done, and manipulation in order to study something, right? That manipulation can only be done on modern systems. Why? Because it involves process of inducing mutations in order to study the phenotypic changes related to the gene mutation. It is ethically not allowed, and more so, it is technically not feasible to induce mutations in humans. Even if you could induce mutations in a human, how will you, like, how long will you need to study them, right? The lifespan of a mouse is around 2.5 years, sometimes even 3, right? So in 3 years, you can study the mouse through its entire age, right? In case of humans, even if you did introduce a mutation, say there were no ethical boundaries and you did manage to induce a mutation, and that mutation is such that it's causing a neurodegenerative disease, which is a late onset disease. Most of the neurodegenerative diseases are late onset. So say this is a late onset neurodegenerative disease, and when you actually did this particular or induce this particular mutation, you were say around 40 years of age. And that particular human, because there has been a global or conditional mutation that has been introduced, might have been a embryo at that time, right? So you will not be able to live until that person actually reaches the age wherein they get neurodegeneration, right? And it will just technically not be feasible. Then again, working on any model system also requires a great deal of ethical clearance. It is not just that we need ethical clearance to work with humans. Even if you want to work on mice, if you want to manipulate even a cell, if you are manipulating the DNA in any way, you need a lot of ethical clearance, right? Also, why I say that for studying purposes or for phenotypic changes, we cannot look at cells. Why did I mention that at humans? In humans, why did I mention that specifically was that there are gene therapies that people are trying now, right? So gene therapy will also kind of modify your genetic makeup, but it is meant as a therapy, right? It will revert your current state to a normal functioning state. Right, so that is why it is not really considered a manipulation, because therein you are regaining your normal state, and that itself is a very big challenge. Like, not many gene therapies have been successful in patients. Over here, I have included one of these examples from EMBO reports. It was a recent example published in 2022, wherein they had basically modified, they had basically taken SARS-CoV-2 clinical strain, right? And what they had done was they obviously knew the sequence of this particular virus, right? Viral genome we are aware of. After this, what they have done is that they have synthetically induced some mutations in this viral genome. And they wanted to see that if we induce these mutations, what kind of outcomes will we see in a model organism, right? For example, if we induce mutations and if we still infected them into the mouse, for example, how will it different from how will the disease of COVID be different in this particular mutant if this particular mutant is introduced into this mouse as compared to the normal clinical strain that was available, right? So it helps in even predicting what kind of changes mutation might bring about in a model system, or eventually which could be extrapolated into humans also, right? So it was basically an attempt to study different kinds of mutations and how basically lethal or fatal they could be if that mutation was brought about in a virus and that would affect a particular organism. Okay.
So my doubt is,
Yeah.
you are trying to tell that reverse genetics we should not try in experiments. Is it like that? Because it is not ethically correct, inducing mutation.
No. I did not say it is not technically correct. I said it was technically not feasible to do in humans. Because somebody asked, why is it done only in model systems? It is technically feasible because, for example, this is one of the latest examples. EMBO is a very good journal, and this particular group, Millard et al., what they have done is what I explained finally. And this is a reverse genetics approach only because they knew the genetic makeup of that viral particle. They manipulated that genetic makeup also, and they wanted to check what outcome will that manipulation bring. So it is a perfectly good system. The only difference between reverse genetics and forward genetics is what kind of applications they are utilizable in, right? The reverse genetics might be a very good model system in, might be a very good approach in some kinds of studies, while it cannot really do genetic tracing, right? For example, if I said that I have a family history of Alzheimer's and I have the medical records of my family also, and can you through reverse genetics tell which gene is causing this particular disease in my family, which gene is making us more susceptible for having Alzheimer's disease. So no, I can't do that because this approach is not fit in that particular hypothesis, right? For that particular hypothesis, you need forward genetics. So, but this is a very good approach when you are manipulating a gene and you want to see its outcome. It is perfectly feasible and perfectly doable, and it is helping the research advance. Okay. So there was a talk about one gene, one enzyme hypothesis during one of the history lectures. I think the introduction lecture 1 or lecture 2. Right, wherein this particular term was used, one gene, one enzyme hypothesis. So, let me just explain what this hypothesis was and who basically proposed it. So, Beadle and Tatum, you remember the people who worked on Neurospora, they basically introduced this particular hypothesis and they also received Nobel Prize in Physiology and Medicine in 1958. Right, this hypothesis was provided in 1941 and they received a Nobel Prize in 1958. And what this hypothesis actually said was that if there is one gene, it will make one enzyme which will be responsible for one particular function. Right? For example, they were studying this ornithine cycle in which they said that the precursor of ornithine needs a particular enzyme for it to convert into ornithine. A second enzyme is responsible for ornithine's conversion into citrulline, and a third enzyme is required for its conversion into arginine, which is the final product that that Neurospora wanted. Right? And they said that these three enzymes that are involved in this process, all these three enzymes have three different genes responsible for production of these enzymes, right? And they basically proved this theory based on their experiments in neurospora. But my question is that from what discussion, from your previous knowledge and from what discussion we have had today, do you think that this is a correct hypothesis?
No.
Yeah. So just a few slides back, we were talking about alternate splicing, right? What was the role of alternate splicing in the first place? The role of alternate splicing was for one gene to be able to form various types of proteins, or maybe even 2 or 3 different types of enzymes, right? So that means that one gene might not just be responsible for formation of one enzyme, right? In case of neurospora, they could prove this experiment because, like Mendel, they were also lucky that they chose those traits and a perfect system wherein this thing was actually true, right? In Mendel's case, Mendelian laws are true in monogenic disorders, where one gene is responsible for one trait. But in a polygenic case, Mendel's laws are not true, right? Similarly, in this case, where one gene was actually responsible for one enzyme, they could actually prove this theory. But it is not the case always, right? How is this hypothesis incorrect? Because it states that the major function of the gene is to form an enzyme that helps in metabolism. They also mentioned that the function of the enzyme was to help in metabolism and other body functions, right? However, we also know that genes encode proteins other than enzymes, such as surface receptors, structural proteins like collagen, elastin, etc. Another thing that they said was one gene can code for one enzyme, while one gene can code for more than one enzyme. This phenomena is possible due to alternative splicing that takes place after transcription, right? Okay, I think this might be the final discussion topic I have, which was epigenetic modifications. So since there was one lecture that was dedicated specifically to epigenetic modifications, I also wanted to introduce some of the modifications that, like, even if you do not know how these modifications happen, because that is a very long lecture altogether, you should know at least what kinds of modifications exist, right? So what are some types of genetic modifications? In this particular illustration, again, I could find a few of them. One was DNA modifications like DNA methylation, right? Apart from DNA methylation, there could also be DNA hemimethylation, which is like an intermediate state between methylation and demethylation, right? Then there could be multiple histone modifications that are present, such as acetylation, methylation, phosphorylation. There was one other that we read earlier, that was ubiquitination or chromatin remodeling based on ATP dependent chromatin remodeling. So these could be other epigenetic modifications that are present. A third one over here is mentioned as miRNAs, right? So miRNAs are microRNAs. So what do these microRNAs do? These microRNAs help in silencing the activity of certain genes, right? So whichever gene they will be complementary towards, they will go and sit on that gene and they will not let it get transcribed. So they will stop its activity or stop its transcription. So miRNAs are also kinds of epigenetic modifiers. Why? Because these miRNAs or microRNAs, or even siRNAs and non-coding RNAs, for instance, what I was explaining earlier, that non-coding RNAs are also epigenetic modifiers because they are regulating the proteins, the gene expression, right? So what all these things are doing is either they'll go and sit on the gene, or they will not help the recruitment of the transcription machinery, something like that. But they are not altering the sequence of the gene or sequence of the DNA, right? So that is why they will be epigenetic modifications and not genetic modifications or mutations. Okay. Okay. This might be my last slide, I think. I just revealed the answer, but okay. Which one is the smallest human chromosome? Since I already came to that slide earlier. Yeah. So it is 21. Okay. So a lot of people believe, and correctly so, because earlier when this karyotype was given, that is when the Jimsa staining of the chromosomes were done and they were arranged according to their size, right? We had not yet come to the genetic sequencing of these chromosomes. We had just visualized them and arranged them according to size. Okay? In that, according to size, when they were arranged from the largest to the smallest, right? And then the sex chromosomes. The autosomes were arranged from the largest to the smallest. And in that, while we were arranging that, we said that chromosome number 22 is the smallest because visually that is what people could make up, right? Once the genome sequencing was done, it was found out that among the autosomes, the smallest sequence is of chromosome number 21. And now this is basically designated as the smallest human chromosome. Now, another question that I would like to pose is it just the smallest human autosome, or is it also the smallest human chromosome altogether? Like, when we talk about both the autosomes and the sex chromosomes, is it still the smallest? Okay, so the answer to that is yes. So a lot of people still did believe that Y chromosome is the smallest chromosome, right? So since Y is a sex chromosome, it is generally not brought into discussion when we are talking about autosomes, which are actually the ones that have been arranged according to size. But when the genome sequencing was done, it was seen that even Y chromosome is larger than the chromosome number 21, right? So this is indeed the smallest human chromosome. When we are talking about both the autosomes as well as the sex chromosomes. Okay. I keep saying that it is my last slide, but I am not arriving there. Sorry. So, again, this was one of the questions put up on the discussion forum, which said, what are hits? Right? So, and I went through the study material, and I basically remembered that during the talk of metagenomics, basically, this word has been used, that is hits. So, it is basically a bioinformatics term. So, I myself am not very knowledgeable about it, but in layman's term, I can explain what it is, right? If we expand this term, that is hits, it is hyperlink induced topic search, right? If we talk about it in layman's terms, what it does is that it is an algorithm which gives us, like if we make a Venn diagram, right? We talk about people at IIT Kanpur is this particular circle, and people doing PhD is this particular circle. Okay? Now this intersection between the two circles gives us people at IIT Kanpur who are doing PhD, right? So this particular circle, whatever would lie in the circle would be a hit when we search using I want people who are doing PhD at IIT Kanpur. It will look into these two databases, an algorithm will look into these two databases, and it will give me the results of whatever is lying between them, which is overlapping between them, right? This would be my hit. So in layman's terms, that is what I can explain. Also, I have kept an example for this. For example, if I search on PubMed, I did the search wherein I wrote human proteins and I obtained 4,277 results, right? Then I looked for yeast proteins and I obtained 1,71,503 results. Thereafter, what I did was I wrote them together. Like I wanted to search publications that talk both about human proteins and yeast proteins, right? So I basically linked the publications that I was the normal searches that I was doing earlier, and it gave me a result which has 442 studies, which basically talks about both human proteins and yeast proteins. So it has basically kind of given me 442 hits. And now I will obviously have to manually go through these hits in order to see whether or not it is something that is relevant to what I wanted to search in the first place, right? So that still has to be done manually, but it has basically narrowed down my study from either 4,000 something or almost 2 lakh results. है ना? Now I just have to go through 400 something results. So that was it. That was finally my last slide. And thank you for attending the session. Just a moment. I'll stop sharing my screen.
Thank you. Your class was nice.
Most welcome. And I have taken note of these two questions that I have on top of my mind. One was a request by Balaji that I basically, what was your request that the current, as a practitioner, what would be the current feasible techniques that you would look at if you had a person's genome data to identify a particular gene that would be causing the cause of the disease, right? One was this question, and the second one was whether or not the exon positions are interchangeable during splicing. So these two things I will definitely check on my part, and I will let you guys know next week. And like my class, my session will constantly run on this particular time only. That is 4 to 6 every Sunday for till the end of your 4 weeks, 4 weeks course. Also, for those of you that have joined later, I had basically conveyed that most probably Sir will also be taking 2 sessions. One will be on 2nd of September, the other will be on 16th of September. That is what had initially been discussed. I am not sure if you have not received an announcement as yet, so it might be subject to change. But maybe you can look out for that, because for today's session, there was no real reminder that was sent. So don't miss Sir's session at least. I think it will be nice. It will be held on 2 Saturdays from 4 to 5 p.m. If the schedule remains the same. So that is all from my side. Thank you so much. My battery is dying, so I will basically sign out. Thank you for attending. Hope to see you in the next class.
Thank you.